Hello everyone, welcome to my channel, and today this video is going to be about membrane of carbohydrates. So you see, this is the updated model of plasma membrane. Okay, so illustrated lipid raft here, and the interaction of transmembrane protein. Okay, these all are the transmembrane protein with the underlying actin cytoskeleton. Okay, this is the actin cytoskeleton. The plasma membrane of eukaryotic cells contain carbohydrates. Okay, this is the carbohydrate chain which is binds to the protein with the covalent bond. So the carbohydrate concentration, it depends on the species and cell type. The carbohydrate content of the plasma membrane ranges between 2 to 10% by weight. There are two kinds of carbohydrates. First is glycoproteins and glycolipids. Okay, so this is the glycoprotein, that proteins, carbohydrates binds with the protein and form the glycoprotein, and same is the oligosaccharides of carbohydrates binds with the lipid and forming the glycolipids. So what is a glycosylation? Okay. So glycosylation is attachment of carbohydrates molecules, for example oligosaccharide, to specific amino acid. For example, this modification affect protein stability. Okay, second one is localization and function. So the linkage, two types of linkage that join sugar to be polypeptide chain. It can be end linked. Okay, end linked, that means end glycosidic linkage, and O linked, that is O glycosidic linkage. In the end linked, that means end glycosidic linkage, it's attached to the aspargine residues. Okay, this is the carbohydrates, and this is the amino acid attached to the aspargine. Okay, aspargine, this is the amino acid, and form the end glycosidic linkage. Okay, in contrast, this is the N-acetylgalactosamine galactosamine and serine and threonine. Okay, the glycosidic O glycosidic linkage formation in this serine amino acid and N-acetylglucosamine. And another amino acid is threonine. Okay, so X indicate it's H, that means this is the serine amino acid, and H suppose indicate the CH3, that means the threonine amino acid. So the more than 90% of the membrane carbohydrates, okay, covalently linked to the protein to form glycoproteins. Okay, this is also second one structure of this. This is a plasma membrane. This is the intracellular domain of protein, and this extracellular domain of protein. Okay, so this is the amino acid sequence and binds to the oligosaccharides with the end linked glycans, and this is the serine amino acid or threonine. You can say the threonine or serine. Okay, so this is a amino acid sequence of proteins binds with the O-glycosidic linkage. So all of the carbohydrates of the plasma membrane faces outward into the extracellular spaces or extracellular spaces. The carbohydrates of intercellular membrane also faces away from the cytosol. For example, endoplasmic reticulums that contains the plasma membrane, they also have the carbohydrates which is faces to the cytoplasm. Okay. So carbohydrates of glycoprotein present is a short branch hydrophilic oligosaccharides, typically having fewer than about 15 sugar per chain. Okay, so oligosaccharide, for example, this is the oligosaccharides that contains the fewer than about 15 sugar per chain. Okay, in contrast to for this is the 15 sugars of oligosaccharide, but in contrast to most high molecular weight carbohydrates, okay, such as glycogen, starch, and cellulose, which are polymer of the single sugar, the oligosaccharide attached to the membrane protein and lipid can display extensive variability in the composition and structure. Okay. So most important things is even the same protein can display different chain of sugar in the different cells and tissue. So oligosaccharide may be attached to several different amino acids by two major types of linkage. This is the N linkage and O linkage. Okay, so here it is the protein. Okay, as you know, the protein is synthesized from the rough endoplasmic reticulums. As each protein is synthesized in the rough endoplasmic reticulums, it becomes inserted into lipid bilayer. Okay, this is the inserted into the lipid bilayer in a predictable orientation determined by its amino acid sequence. the first the what happened, the carbohydrate chain. Okay, so this is red, it shows the carbohydrate chain is which are first added in the ER. Okay, so this is the added into the ER, and then provide a convenient way to assess membrane sideness because they are always present on the cisternal side of the cytoplasmic membrane. They transport the protein transfer the Golgi apparatus, Golgi complex, okay, which becomes the exoplasmic side to plasma membrane following the fusion of vesicle with the plasma membrane. Then they fusion with the vesicles and they attach to the plasma membrane exterior sides. Okay. So the remaining carbohydrates is currently linked to the lipid to form glycolipids. Okay, so this is which were we discussed in my previous video, lipid structure plasma in the which ganglioside lipid has oligosaccharide chain bind to the phosphate head of the lipid. So let's consider one of the example. Okay, for instance, uh, okay, so carbohydrates can determine the person blood group also. So the carbohydrates of the glycolipids of the red blood cells plasma may determine whether the person blood type is A, okay, A, B or O. Okay. So for so the enzyme, okay, this is the main reason is the enzyme. For example, if person having A blood group, okay, so the basic difference between this like A antigen, okay, this is the lipids which binds the oligosaccharides. Okay, lipids binds to the oligosaccharide, lipids binds to the oligosaccharides, either protein or lipids. Okay. So the first O antigen, okay, if person having O antigen, blood group antigens, so this is the oligosaccharide chain. Okay, so there is the GLU, that is glucose, GLA, that is the galactose. Okay, and the galactose, okay, N-acetyl glucosamine, okay, and N-acetyl glucosamine. Okay, so galactose and fucose, for fucose. Okay, so here it is no side, this side chain, it has nothing extra sugar. Okay, but here you can see A and B, there is extra sugar. Okay, so if person have A blood group, okay, that is this red blood cells having oligosaccharides same is the O, but there is one different, that is the galactosamine. Here is a present, that means A antigen, A blood group. In B blood group there is one more, okay, galactose is present. Okay, so this is the basic difference between blood group of antigen. This is determined by the carbohydrates which are present in the red blood cell, which commonly binds to the protein and lipid. Okay, so this is all about today's videos. Okay, I hope this video will helpful you. Okay.